Okay, Mario, we are live now. Good luck to everyone.
Thank you very much. So maybe the speaker that are not intended to have the presentation should turn off the camera in order to do not overload the communication, please. I will do the same. Thank you very much. So, Fiammetta, you can start. Thank you very much.
Well, thank you, Mario. Thank you, Mario. So, I'm Fiammetta Berlinguer, the coordinator of the PhD program in veterinary science from the University of Sassari. And I want to welcome all to the first edition of the Research and Communication Contest for the student of the program in veterinary science of the University of Sassari. Today, this morning, our PhD student answer a question from experts from an international jury. The first classified of each year will be awarded €500 to be used. Please switch on your mic, switch off your microphone. Again. So the first classified of each year will be awarded with €500 to be used to carry on their research. The event will be in English, will be streamed live on the Facebook page of the Department of Veterinary Medicine of Sassari. Strong oral communication skills are always valued, and we'd like our PhD student to get more public speaking opportunities than the most. Through conference calls, poster presentation, and also through teaching, you will learn to feel comfortable in front of a larger audience, engage them, and present complex ideas in a straightforward ways. If you practice speaking and writing for a variety of arenas, you will feel more comfortable when it really matters. And so having speaking opportunities is like an exercise to gain confidence. One day, for example, you will win a teaching award or be recognized as the best speaker of a conference. And this will be a concrete way to prove your public speaking skills. So today, pay attention, you will have just 3 minutes to deliver a compelling presentation on your research. And you can use only a single static slide. This exercise will help you develop skill in translating your research and its impact. And this is essential for many purposes, for example, for funding proposal, for transitioning to employment, or also to generate interest and awareness in research and academic research especially. And of course, to improve your communication skill with conference in a variety of audience. So please today get involved, do your best, and good luck. But before giving the floor to Professor Mario Giorgi from our PhD board, who will chair the event, I would like to introduce our international jury. Professor Maria Teresa Paramio is from the Department of Animal and Food Science of the University of Barcelona. Her research line mainly focuses on small ruminant reproduction, particularly the mechanism that allow the oocyte to gain the developmental competence to improve in vitro production of good and viable blastocysts and embryos. Professor Paramio also participates in the PhD program in animal production from the University of Barcelona. And this program has gained the prestigious mention for excellence from the Ministry of Spanish Ministries for Education. Our course has an agreement with the PhD program in animal production from the University of Barcelona for a double degree, for a PhD double degree. So please take aware of this excellent learning opportunity and contact me for any further information. Professor Taewon Kim is from the College of Veterinary Medicine of the Chungham National University in South Korea. Professor Kim's research lines mainly focus on pharmacokinetics and pharmacodynamics in domestic animals, and this is a key aspect in the development and enhancement of the veterinary practice. Our course also signed an agreement with the CNU to develop an international PhD student exchange program in veterinary science between the two universities. The Chungham National University, the College of Veterinary Medicine of the Chungham National University is really a gold standard in all the veterinary practice, especially related to the internal medicine and surgery. So once again, contact me. The University of Sassari has specific funding for extra EU international student mobility. So be aware also of this important opportunity. Professor Chadi Osri is from the Lebanese University. with which we have an ongoing and long-lasting collaboration. Professor Osri works in several aspects of veterinary science related also to the peculiarities of the of his country, and also with the food quality of animal origin. From several years, as I just mentioned you, our course has an agreement with the Lebanese University, and we reserve each year position to students from the Lebanese University. And this collaboration over the years has fostered internationalization of our course and also the cooperation in the Mediterranean area. So they form the international jury, and they will evaluate your speech, giving a specific score in one of these categories, which are presentation quality, visual aids, and content. They will have a total of 20 scores for each oral presentation. So 5 scores will be granted for presentation quality in terms of English and oral skills, communication, and timing. Sorry, so please be aware of staying the 3 minutes. 5 scores to the visual aids. You should be clear and concise, well developed and appropriate. And 10 score to the content, of course, the quality of the research in terms of background, result, conclusion, and so on. The award of the winners will take place after the end of the oral presentation. After that, I will also give an overview of the open call for the 37th cycle of the PhD program. So more or less that's all. Once again, good luck to everybody. And I will give now the floor to Mario, which will chair the event and start with the oral presentation. Good luck and see you later.
Thank you very much, Shametta. And also I agree with you in the thanksgiving to the expert member board. And I don't want to lose time because we are already behind the schedule. So I would like to ask Dr. Akle Taufik to turn on the camera, please.
Good morning.
Good morning. So Dr. Akle will present us a research titled Tick-borne disease in European mountainous wild ungulates, a systematic review. So yes. Dr. Akle, the floor is yours. The time is starting. Good morning.
Okay. Good morning. Okay. Good morning. So tick-borne diseases in wild ungulates, their status is not always clear. And these diseases can sometimes be transmitted to domestic animals and humans. For this reason, we decided to conduct a systematic review investigating the prevalence, geographic distribution, clinical description, and reservoir host status of a few tick-borne diseases in European mountainous wild ungulates. For this purpose, we conducted a database research using for keywords the 3 animal species of interest, which are mouflon, chamois, and ibex, combined with the names of the pathogens of interest, Rickettsia, Theileria, Babesia, Anaplasma, Borrelia burgdorferi sensulato, and Coxiella burnetii. From the data that from the publications that were included in our systematic review, we were able to draw this summary table. As you can see, all the studies had taken samples of either tissue or ticks collected on wild ungulates. As you can see, all the pathogens, all the tick-borne pathogens that were investigated in all the animal species were found, to the exception of Theileria species, which was never investigated in ibex, and Rickettsia species, which was only investigated once in chamois and ibex and was not detected. The only pathogen to have been described clinically was the Babesia species, which was done on 4 occasions in chamois and a single occasion in an Iberian ibex. These animals had typical babesiosis symptoms of babesiosis, such as anemia, liver and spleen hypertrophy, and pyroplasmic inclusion bodies inside of the erythrocytes. And the chamois were positive on molecular testing, while the ibex were seropositive. From this data, we were able to draw also these unique maps, which show the distribution of the tick-borne pathogens in the European mountainous ungulates. As you can see in these maps, unfortunately, there's a lot of what I would call virgin areas. These areas have populations of European mountainous ungulates that were never investigated for these tick-borne pathogens. Also, of course, we do realize that these virgin areas do not signify the absence of studies, but rather that the data produced was kept inside of the drawers. And this is actually the aim of our systematic review, and that is to highlight the blank spots and encourage wildlife biologists and researchers to publish their data in order to allow the scientific community to have a fuller and clearer view of the epidemiological status of tick-borne pathogens in European mountainous ungulates. Thank you.
So thank you very much, Dr. Akle, perfectly on time. And now is the question time is open.
Okay.
So has the member board any question? Can you please unshare your screen?
Yes, of
Yes,
course.
good morning. Good morning, Tufi. I hope that you are fine, long time. I have a question, firstly.
Of course.
In the literature review, did you find data from other countries and European country where you work? And is there a big differences in the results about your data and other countries, if you found any?
Okay, so first to clarify, our systematic review is focused on European animals and the three animal species that I mentioned, the chamois, mouflon, and ibex. So all publications that were not within these inclusion criteria were discarded. But of course, there were a lot of studies in other countries. I was just updating my data a few days ago, and I found a lot of studies in Yukon in Canada, some in China, of course, and there's from all over the world where there are wild ungulates. There has been also a lot of studies, yes. But we did not include these because they were not they do not meet the criteria of inclusion.
Okay, sure. Thank you. Thank you.
So thank you very much. I guess thank you very much, Dr. Akler. I guess it's time to proceed with the next speaker.
Thank you.
And thank you very much, Professor Chadi, for your question. So the next speaker is Jelwan Rachel. She will present a research titled Seroprevalence of Toxoplasma gondii infection in cats population in Beirut and Mount Lebanon, Lebanon. So, Dr. Jelwan, the floor is yours.
Good morning. First, toxoplasmosis is a worldwide protozoan disease caused by a parasite called Toxoplasma gondii. Only Felidae are known to be definitive host, while almost all warm-blooded, including a human, are intermediate hosts. In 1983, 78% of cats in Beirut had the Gondii antibody, while among Arab countries, Lebanon represents the highest seroprevalence in T. gondii antibody in pregnant women. So our objectives is to provide, after 35 years, an updated estimate about the seroprevalence of T. gondii antibodies in cats in Beirut, and to assess for the first time the seroprevalence in Mount Lebanon, and to assess the affecting factors. So a blood collection were randomly collected from 104 cats in 2018 and 92 cats in 2019. An ELISA kit were used according to the manufacturer instructions. And the questionnaire was filled, including the following factors in the table, such as governorate, gender, age, origin, lifestyle, hunting behavior, living with other pets, breed, nutrition, reproductive status, and the presence of concurrent disease. As a result, we obtained an overall seroprevalence of 20.2% in 2018 and 20.7% in 2019. A significant difference was found with the cat's lifestyle in 2018, while no significant difference was found in any of the studied variables in 2019. So in general, the observed factors do not seem to be strongly associated with the disease, which is confirmed by the data of 2019. And then in conclusion, there is a decrease in the overall seroprevalence in cats comparing to the previous study, which point on the importance of searching other causes of human contamination in Lebanon, and further investigation about relationship between factors are needed. Thank you.
Thank you very much, Dr. Jelwan, in order to stay to respect the time. So the question time is open. There is any question from the audience and from the panel board member, please.
I don't think so. We can go.
So, yeah, so thank you very much, Dr. Jelvan, for your presentation. And now it's time for Dr. Meloni Maria Pia. She will present a study titled, "Can Protective Culture Improve Sheep's Milk Cheese's Safety?"
Good morning.
Dr. Meloni, good morning. The floor is yours.
Thank you. Can protective cultures improve sheep's milk cheese safety? Protective cultures can be regarded as an interesting opportunity to minimize the risk of persistence or development of Listeria monocytogenes in ready-to-eat food like Pecorino Sardo PDO cheese. But to introduce protective cultures on the production process of PDO cheeses, it's essential to use only autochthonous microorganisms to respect the specifications provided. But commercial protective cultures are not autochthonous, and they are composed by mesophilic strains like Lactobacillus plantarus. And some cheese-making processes are designed to promote primary fermentation of thermophilic strains. For this reason, my study had two main goals. The first one is the selection and identification of autochthonous thermophilic lactic acid bacteria that could be used as a protective cultures for Pecorino Sardo PDO production. And the second one is the in vitro comparison between commercial protective cultures and autochthonous lactic acid bacteria antimicrobial activity against Listeria monocytogenes. I isolated 220 bacteria strains from raw sheep's milk, and 74 of them were selected as a presumptive thermophilic lactic acid bacteria. Species identification is carried out using 16S RNA sequencing. The inhibitory activity of commercial protective cultures and autochthonous lactic acid bacteria against 50 Listeria monocytogenes strains and their ability to produce bacteriocin was carried out using well-diffusion assay method. Commercial protective cultures on the left in these pictures and autochthonous lactic acid bacteria on the right were inoculated in their specific media and in scotta, the milk waste residual from ricotta cheese production, to investigate their adaptability on the dairy products. In this picture, I compare the inhibitory activity in vitro of these two kinds of bacteria. On the left, we can see the inhibition zone diameter of commercial protective cultures that goes from 1.6 to 2.2 centimeters. On the right, we can see the inhibition zone diameter of autochthonous lactic acid bacteria that goes from 1.1 to 1.9 centimeters. There is a little different, but it's absolutely normal because commercial protective cultures were created to have a specific antibacterial activity and autochthonous lactic acid bacteria not. For this reason, we can confirm that their antibacterial activity is absolutely comparable. And the autochthonous lactic acid bacteria may be considered as a good candidate for a future creation of a new protective culture for Pecorino Sardo PDO cheese. Thank you.
Thank you very much, Dr. Dr. Meloni, for your nice presentation concerning the cheese, and question time is on. Any question from the expert panel member?
Thank you for the presentation. Just curiosity. So I'm quite dumb at cheese production. I should turn on the video. Yeah. Hi, hello. I just wondering that if we use lactic acid bacillus, can the LAB can affect cheese quality or not because of the acidity?
Yes, I tried to inoculate the lactic acid bacteria in milk and in scotta, the residual from ricotta cheese production. They demonstrated a good a good growth of pH decrease, and it's be a good chance. Because it's important to have a good acidification in cheese. For this reason, we can compare their activity to the starter activity, and we can use dairy in main activities with starters and other microorganisms during the cheese production. It's a good result for the moment.
Okay, then thanks a lot.
You're welcome.
Thank you very much, both. And let's proceed further. Now it's time for Dr. Zecchi Valentina. She will present research titled Strain Elastography of Injured Equine Superficial Digital Flexor Tendon, SDFT, a reliability study. So, Dr. Zecchi,
good morning,
turn on the
everyone.
camera, please.
Can you see me?
Yeah, here you are. Thank you very much. The floor is yours.
Thank you. So, sports horses, oh, sorry. Can you see the screen?
Not so far. Don't panic. Try to fix it. Yeah, now it's clear.
No.
Not now. Maybe you can leave the slide in the
perfect.
Not showing presentation. Yeah, so we can read clearly. So don't worry.
Sorry.
No worries.
So, sport horses are particularly subject to tendon injuries, and this is a huge issue due to the high recurrence rate and the time needed for complete healing. Two-dimensional ultrasonography is commonly used to diagnose and monitor equine tendinopathies. However, it is not able to assess the mechanical properties of tissues, not to predict injuries or detect abnormal signs in tendons over 5 months post-injury. On the contrary, strain elastography is an ultrasound-based technique that assesses the mechanical properties of tissues and gives a relative representation of elasticity. Early diagnosis of tendon injuries and long-term monitoring of the healing process are key to equine practice. Thus, an accurate method is needed for analyzing and interpreting the images obtained with strain elastography. So, in our project, the aims were mainly two. The first one was to demonstrate the interoperator reproducibility and the interoperator reproducibility of manual measurement of elastograms. These were obtained from injured superficial digital flexor tendon, also called SDFTs, in horses in training or racing programs. The second aim was to perform a validation of this manual measurement method by comparing it with an external software that we choose to be ImageJ because it's a free available software for everyone. Regarding material methods, 20 elastograms of injured superficial digital flexor tendon in long axis view were randomly selected from the most representative videotapes and then analyzed by 2 different operators after an acute injury was diagnosed with ultrasonography and then assessed with elastography. The analysis was first performed manually with the tools available on the ultrasound unit and then analyzed with the external software, as you can see on the image on the left side of the slide. Despite its subjectivity, manual measurements proved to be repeatable and reproducible, obtaining an excellent inter-observer repeatability with an intercorrelation coefficient of 0.9 and good inter-observer reproducibility with an intercorrelation coefficient of 0.8. In addition, the results obtained with the manual method matched those obtained with the external software ImageJ, obtaining a good agreement with an ICC of 0.8. To conclude, we believe that this demonstrates that manual measurement is a feasible and accurate method and that it could be considered as valid as the objective software assessment. As an accurate interpretation and measurement of lesions is fundamental, it could thus be used to evaluate tendon healing in the future. Thank you very much. I'm sorry.
Thank you very much to you for your nice presentation. And now it's time for question. Any question from the panel member board? So it looks like there is no question, but I have a question for you. I'm not a clinician, I'm not a vet, but the question is extremely, stupid. Can this method be used also for other animal species, like dogs, cats, or?
Yes, of course. We made some trials on dogs too, and we made also a poster that was presented on an international congress in Spain with the collaboration of Dr. Alicia Barbero Fernandez. So we had preliminary results, and it could be really nice if we could continue to study this technique, because in veterinary there are really few studies. In fact, in order to improve my knowledge on the topic, I need to attend an international congress in human medicine.
Okay, thank you very much indeed. So let's go ahead with Dr. Irene Sartini. Dr. Sartini is going to present a research titled Pharmacokinetics of doxycycline in the goods. So, Dr. Sartini, please turn on your camera.
Good morning.
Good morning to you. Okay, we can see your slide and the floor is yours.
Okay, thank you. So it's generally accepted that geese have been one of the first animals to be domesticated, and thanks to their ability to adapt to different climates, the commercial goose production became important in different countries in Asia as well as in Europe. Unfortunately, as many other avian species, they can be affected by different pathogens such as Mycoplasma species that cause reduced growth, reduced production, increasing mortality and treatment cost. Mycoplasma species is sensitive to different antibiotics, including tetracyclines. That's the reason why we investigated doxycycline, a semi-synthetic tetracycline, which shows an excellent effectiveness against Mycoplasma gallisepticum in vitro. Doxycycline is used in the treatment and prevention of different kind of infection in poultry, but there are no data available in geese yet. What I mean is there are missing data on its absorption, exposure, elimination, and consequently on the optimal dosage, which cannot be extrapolated from other species. It goes without saying that a pharmacokinetic assessment was needed. For this purpose, 10 geese were enrolled in a two-phase crossover study with a washout period of 2 weeks. All the animals were treated intravenously and orally at 20 milligrams per kilogram with doxycycline. Blood was collected at a fixed time point after the administration, and the drug was quantified. The most relevant features of the drug were found to be a long elimination half-life and an incomplete oral bioavailability. But what about efficacy? Doxycycline is a time-dependent antibiotic, and the pharmacokinetic pharmacodynamic breakpoint, which has been proposed to predict the success of doxycycline therapy, is the duration of plasma concentration exceeding the minimum inhibitory concentration value. This index should be at least 54%, but in the present study, it was 71%. So it suggests that an appropriate therapeutic efficacy has been reached in this disease against Mycoplasma gallisepticum. But now, the current dosage approved of doxycycline in poultry is 10 or 20 milligram per kilogram of doxycycline administered daily for 3 or 5 days. So the question is, what will be the predicted plasma concentration after a multiple dose administration? Can the pharmacokinetic pharmacodynamic breakpoint be reached? From the in silico simulation that we performed, the answer seems to be yes. The lower dosage, 10 milligram per kilogram, seems to provide plasma concentration enough high in order to obtain an appropriate clinical efficacy in this case. Thank you.
Thank you very much, Dr. Sartini, for your presentation. Now it's time for question. There any question?
Yeah, just one question. Is this antibiotic forbidden for the market? Yeah, in order to the meat consumed?
No, it's not banned. It can be used also in the EMA guideline can be found. It's generally used in as a powder to solubilized in water, so as a water medication administration. And in fact, in this study, we didn't focus on the possible limitation that we can observe in the field, because of course there is the intervariability between subject, between the daylight and the cycle. So, but yeah, it can be used.
Okay, thank you.
So thank you very much for the question, for the answer. So let's proceed now to the 35th grade. Now I ask Dr. Fahad, that is already visible there, to present his study titled, How Can Vultures Contribute to Cystic Echinococcosis Control? So, Dr. Fahad, the floor is yours.
Okay. Good morning. Cystic echinococcus is endemic in Sardinia, meanwhile deserving attention. Despite various attempts, the control of disease was unsuccessful in the region. This is mainly attributed to many factors. However, high cost of incineration, free access of dogs to unattended carry-ons are major behind the spread of CE to the livestock. A wildlife conservation program in Sardinia entitled Life Under Griffon Wing aimed to expand the Griffon vultures population by establishing supplementary feeding station and providing sustained supply from the dead carcasses. Griffon vultures are uniquely specialized in removing carcasses efficiently and swiftly. We aim to ascertain these natural foraging habits in controlled disposal of carcasses, thereby breaking the life cycle of CE in Sardinia. A total of 37 feeding stations were established. Feeding stations were in closed space, surrounded by an electric fence with no chance of access to the canids. The feeding stations were associated with sheep, goat, cattle, equine farms. The carcasses include 255 individuals from sheep goat, 171 individuals from the cattle, and 2 from the equines. All the carcasses before feeding to vultures were inspected for sanitation as well as any drug residues. In results, 81,361 kg of biomass was removed during the course of the project, saving 90,041 euro of incineration and 1,054 euro carbon emission costs. The picture is one of the beautiful description between the difference between two of the sites. Green economy is depicting saving lots of high amount of money, as well as the emission of, as well as stopping the emission of greenhouse gases. We extrapolated these results to identify the amount of carrion, infected carrion that was removed during the course of conservation project. So we took into account the recent epidemiological survey in the sheep, which accounts for 65% prevalence. And the result shows that 5,304 kg of infected sheep biomass was removed. We also quantified the amount of the biomass which would be removed in the preceding project, which is kicked off in 2021, and which aims to expand Graph-on vultures to the whole of Sardinia. And we're expecting the double amount of biomass would be removed, which is equals to 1,62,722 kg, and double amount of infected sheep biomass would be removed, further lowering the disease burden in Sardinia. In conclusion, our results underscore the efficient and peculiar characteristic of Graph-on in removing the infected biomass without problem of disease spillover, as well as various ecosystem and economic services. This is one of the pioneer investigation for the control of cystic echinococcus, which can be spread to the other endemic areas. Thank you very much.
Professor Fahad, thank you to you. And now is time for the question. Is there any?
Thanks for the interesting presentation. I have one simple question.
Thank you very much.
Yeah. As far as I understood, so the vulture can help to prevent cystic kind of disease control, right? So
yes.
Is there any negative aspect if the vulture can, manifest it or something like that?
There are some negative effects because some of the bacterial diseases can be spread by the bacterium, by the vultures, such as Salmonella, but they are not. They are not sure about their epidemiological spillover, and these are just like speculations. And furthermore, vultures have some trophic cascades effects on the animals, but not too much. There are reported some of the documented studies that the farmers find the interaction between wildlife vultures and the herbivores, but that is too negligible as compared to the importance of this point.
Okay, then. Thanks a lot.
Okay, so thank you very much to both. Now it's time for Frau Adele, Dr. Frau. Good morning, Dr.
Morning. Good morning.
She is going to present research titled "Can anti-Mulleran hormone predict her productive life in dairy heifers?"
Okay.
So, Dr. Frau, the floor is yours.
Can you see the screen?
Yes, perfectly.
Okay. In literature, different studies indicate that the ovarian reserve that is represented by the total number of healthy follicles and oocytes is positively associated with fertility in dairy cattle. In this study, we hypothesized that the productive herd life could depend on the ovarian reserve, and in particular that a smaller ovarian reserve is associated with a shorter productive life in comparison to herd mates with a greater ovarian reserve. Two fertility markers were used in this study: the AFC, the antral follicle count, that we measured on a random day of the estrous cycle by transrectal ovarian ultrasound, and peripheral AMH, the anti-Mullerian hormone, evaluated using a commercial kit, ELISA kit, a bovine ELISA kit, on a single blood sample. We evaluated both markers on a cohort of 251 Holstein Friesian heifers located in 2 dairy farms in Sardinia, Farm 1 with 135 animals and Farm 2 with 116 animals. Farm records were also analyzed after 2 years to identify culled animals and their age at culling. We found that average AMH concentration was higher in Farm 1 compared to Farm 2. AFC was similar between farms. Also, Farm 1 had higher culling rate and lower age at culling compared to Farm 2. We analyzed also culled cattle, and we found that the number of artificial inseminations from last parturition to culling was higher in Farm 1 compared to Farm 2. In conclusion, with these preliminary findings, we found that heifers with a smaller ovarian reserve had a longer productive life and were culled at a lower rate compared to heifers with a greater AMH serum concentration. An ulterior study that enrolled a larger number of animals is necessary to establish the association between the size of the ovarian reserve and longevity. Thank you.
Thank you very much. So, question time. Any question from,
Yes,
Maria, go ahead.
Yes, I would like to explain a little more about the material and methods. How many and in which moment you take the samples to analyze the anti-mullerin?
We took it only one time in a random day. Also for the antral follicle count, we take both samples the same days. I had this data from the previous study, so I didn't work on this because now I'm working on another project. So I use the same data to have other results. So we take the antral follicle count and the anti-mullerin hormone the same day. So we take both samples the same day.
Mm-hmm. And well, so this is difficult to relate it with the reproductive life, no, just with one sample. But have you shown a correlation between what you see in the ovary with the level or the concentration of the anti-mullerin hormone?
Between the two, yes.
Yes.
Yes, there is a correlation. Yes, are positively correlated.
A positive that it means more small follicles with higher.
Yes, a higher number of antral follicle counts are associated with the higher value of anti-mullerin hormone.
Okay, thank you. Thank you.
Thank you.
Mm-hmm.
Thank you for the question. Thank you for the answer. So now it's time to proceed further. It's now time for Dr. Knoll to turn on her camera. And she's going to present us a research titled...
Hello, sorry.
Oh, sorry.
Yes,
no, is made. Sorry.
Yes.
She's going to present us a research titled Environmental Contamination by Dog Feces in Touristic Areas of Italy, Parasitological Aspect and Zoonotic Hazards.
Yeah. So can you see the screen?
Yeah, I can see perfectly.
Okay. So this research concerns the environmental contamination of urban areas with dog feces and the risk this could pose to animal health and human health. Around the globe, public spaces have been shown to be common sites of fecal contamination, which is concerning given the high prevalence of gastrointestinal pathogens carried by pets. So for this reason, this research aimed to assess the fecal contamination of two of the most important cities on the island of Sardinia, a popular tourist destination, as well as the parasitological risk associated with such contamination. So this was done applying a grid approach, dividing both cities into many equal-sized squares. Then within each square, a 200-meter transect was defined, and the amount of feces for each transect was counted, and 1 in 3 feces were collected for parasitological analysis. So on the left and right, you can see the results, which show widespread contamination for both cities. For Sassari, 956 feces were counted with at least 1 in 90% of the grid squares, and with an average of 4.7 feces per 100 linear meters. And almost 16% contained parasitic elements. The situation in Alghero was somewhat better, with 220 feces and an average of 2.6 per 100 meters. And almost 7% contained parasitic elements. The parasites encountered in this research, which are shown in the center of the slide, mostly have a direct life cycle with a large excretion capacity, thus leading to high contamination risk in areas with dense dog populations, as is the case for Sardinia, where currently over 500,000 dogs reside, or 1 in 3 people. Besides, this number increases significantly in summer, as tourists often travel with their pets to the islands. But fecal contamination does not only affect dogs. The majority of the parasites found in this research, 4 nematodes and 1 protozoa, these are the ones indicated in red on the slide, are known to be able to infect and cause illness in humans as well. So the high fecal contamination reported here does not only represent an animal health problem, but also a significant public health concern, which, besides posing a risk to the residents, also affects the many tourists visiting Sardinia each year. This research has thus shown the need for improved education and sensibilization campaigns in order to stimulate people to properly dispose of their pet feces and to apply proper parasitological care for their animals. So working together, we can go for a safer, healthier, and cleaner Sardinia. Thank you.
Thank you very much, Dr. Knoll. Now it's time for question.
Yes, thank you, Dr. Knoll. My question is, what is the risk for human, or in other way, the question is, how is the risk of human contamination or transmission of this Echinococcus to human?
Echinococcus was not found during this research, which was a very positive note. So, but the main contamination route for humans is when, which happens a lot, in cities, is when you step in feces and the feces particles will stick to your shoes and they will be dragged into your house or into buildings. And there, the most susceptible people are, for example, kids and babies, which are also the most susceptible to parasite infections. And otherwise, you have a higher risk for people that, for example, push a baby stroller, or people with wheelchairs to contaminate the wheels, which will again contaminate people's houses, or even the hands if the wheels of the wheelchair gets with particles of feces.
Thank you.
All right.
Thank you very much for the question and the answer. So thank you very much, Dr. Knoll. It's time for now Meloni, Dr. Meloni Luisa, that is already, turned on the camera. And Dr. Meloni is going to present a research titled Parascaris SPP in Horses of Italy, a large-scale epidemiological analysis in the egg extraction and conditioning sectors. So, Dr. Meloni, the floor is yours.
Equinocariosis is a disease caused by different species of Parascaris. As demonstrated by recent studies in the literature, this parasite can be found up to 100% in foals. However, in Italy, there are no epidemic large-scale studies concerning equinocariosis. For this reason, the present survey aims to describe for the first time the spread and infestation of different species of Parascaris in a large population of horses throughout Italy, using fecal eggs count and, furthermore, identify risk factors associated with Ascaridex shedding. We analyzed about 7,000 horses from over 1,500 stables in Italy. Fecal egg count was performed by McMaster coprological examination modified using a sodium chloride supersaturated solution for flotation with a specific gravity 1.2. A prevalence of 6.3% of the animal examined was found, and a prevalence of 35.8% of the stable showed at least one positive animals. The prevalence of roundworms in foals less than 1 year old is 36.7%. In line with recent studies, foals are considered susceptible to Ascarid infection. In fact, age is one of the main risk factors. This is important because cases of Ascarid-associated colic occur most often in foals between 4 and 7 months of age. Results regarding the animal housing suggest keeping animals outdoors compared to the horses being kept in stables to be an important risk factor for the occurrence of ascariosis, as the literature reported. A statistically significant difference was found between horses reared in the north and in south of Italy, showing an increase in positive in the northern countries. In conclusion, ascariosis represents an health challenge for horses reared in Italy, especially young animals. Furthermore, the pathology in adult animals should not be underestimated because they are an important source of contagion for foals. Finally, housing type, especially outdoors rearing and geographical location, should be implemented as a risk factor with Parascaris monitoring and control programs for Italian horses, with a specific focus on reduction of risk exposure. Thank you.
Thank you very much, Dr. Meloni. It's now time for question. Any? So I have a silly question for you. You said that you have found a difference in contamination between the North and the South Italy. Am I right?
The different contamination, yes, is present in the north and south of Italy, but the parasite present in the fishes is prevalent strongyloids and different all different Parascaris, but the Parascaris in these studies is 2 species, and I don't identify because I use the flotation. And for identify the different species, we use the biomolecular exam.
Okay, thank you very much, Dr. Meloni. It's now time for the last speaker of the 35th grade, Petretto Elena.
Hi, hello.
See you,
Good morning.
Dr. Petretto. She's going to introduce us a research titled Evaluating Genetic Diversity of Sarda breed bats by mtDNA and Y chromosome markers. So, Dr. Petretto, sorry, the floor is yours.
Thank you. Good morning, everyone. Let me introduce our work aimed at the evaluation of genetic diversity and population substructure of 136 Sarda breed bats raised in the island of Sardinia and deriving from 8 sampling sites, here named subregions. After purifying DNA from blood samples, we perform genotyping of 7 SNPs in order to trace patrilineal diversity, and we perform the sequencing of the hypervariable region of mitochondrial DNA in order to trace matrilinear diversity as a source of complementary information. Analysis of Y chromosome data reveal the segregation of 5 haplotypes, and we built a median genetic network here in figure 1. And this haplotype had been already advanced in the different regions of Europe, Asia, and Middle East. And to our great surprise, we found them all in the island of Sardinia. High levels of Y chromosome diversity were observed in the southwest of Sardinia, probably due to the introduction of cosmopolitan breeds to improve meat production. The median genetic network in figure 3 of mitochondrial DNA sequencing from Sarda bats here reveal the occurrence of high variability. And here we also identify different clades already revealed in Sarda female goats, and including clade A11, which is considered to be specific to Sardinia. The median genetic network in figure 4, including Sarda and European, Asia, and African Capri mitochondrial DNA sequences, and reveal the Sarda bats belonging to haplogroup A. And then 3 sequences were in haplogroup C here. In the network, they cluster near the Corsican goats. AMOV analysis support the lack of population substructure for both Y chromosome markers and mitochondrial DNA markers. And our results suggest the occurrence of historical and extensive gene flow between Sarda goat populations from different locations of Sardinia. The integration with the foreign caprine breeds in order to improve milk production may have contributed to avoiding the genetic differentiation amongst Sarda populations. Thank you.
So thank you very much, Dr. Petretto. Is there any question?
Yes, please. Yes, please. I would like to know, I don't know this breed. Is a milking or a meat breed?
Yes, a milking. This goat is for milk production, yes.
Okay, okay.
Yes.
So any of these genetic variation have a relationship with the production of milk?
We worked here only for genetic variation, so genetic diversity. So I think this is a progress. No, I don't know, a new point. New point, yes.
Okay, okay,
yes.
But at average, how is the production of this breed, more or less, just to compare with my goat breeds? Never seen
I don't
your,
know, like,
I would like
I
a
know,
picture here.
yes, yes. And
more or less
like, yes, like Murciana, I think.
For example.
Yes, like Murciana Granadina.
Yeah, Granadina. What is the production of this milk in in a year? Yeah, for kilograms of milk or 800 kilograms of milk? Or
yes, more or less.
Okay.
Yes, more or less.
Okay. Okay. Thank you. Thank you, Elena.
Thank you.
So thank you very much. I have to apologize with you all because, you know, I have some problem to my eyes. You can see me, you know, putting and lifting my glasses. So I guess forgot one speaker for the 35th year, Havko Salka. Is he or she present? No, okay. So, yeah. Are you Havko Salka? Santu? No.
I'm from the 35.
No. So you are from the 36. 36. Yeah. Okay. So now let's go ahead with the 36th grade, and the speaker is Dr. Cavallo Lia. She is going to present. Can Cavallo Lia?
Good morning.
Good morning.
Good morning.
So she's going to present a research titled Species Diversity and Molecular Insights into Phlebotomine Sandflies in Sardinia, Italy, an endemic region for Leishmaniasis. Dear Dr. Cavallo, the floor is yours.
Okay. Can you see me?
Yes, very well.
Okay. Phlebotomine sandflies are a small arthropod of veterinary importance and a serious public health problem. In fact, they are a vector of many pathogens as phlebiviruses and also parasites as Leishmaniasis. This last protozoa has been considered endemic for a long time in Sardinia. However, the information on the distribution and ecology in the island goes back 30 years. Our research would like to update entomological data in the island by the combined use of morphology and molecular taxonomy. Sandflies were trapped from April to November and with two different collection methods, sticky traps and CDC-like traps, installed before sunset for 12 hours in 5 different capture sites where there was different weather and latitude. All phlebotomine sandflies were identified molecularly and morphologically according to the morphometric case provided by Dantas Torres. 513 phlebotomine sandflies belonging to 3 different species, Phlebotomus perniciosus, Phlebotomus versillievi, and Sargentomia minuta were collected. Both Phlebotomus perniciosus and versillievi are proven vector of Leishmanian phantom and may indicate a risk for humans and dogs living in the Iceland. The vector fly season began in May and to end in October, and the peak of activity of the fly vector season was in the summer months, in particular in July and in August, when the temperature were higher, when there was a low relative humidity, and when winds were weak or absent. Therefore, systematic monitoring of this vector in endemic area as Sardinia is the key for development of effective control strategies for vector-borne disease as leishmaniosis, not only in Sardinia but also in Mediterranean area. In fact, the transmission of Leishmanian phantom is abroad. For example, in the northern part of Italy, is an increasing risk. Therefore, for this reason, the use of topical reference.
Dr. Cavallo, you have to conclude because the time is over.
Yes, yes. The use of topical repellent preventive measures are on dogs and cats, for example, permethrin or deltamethrin. Spot on could be effective to prevent the phlebotomine bites and the Leishmanian phantom transmission. Thank you.
Thank you very much. Any question for Dr. Cavallo?
Thanks for the presentation. I have just one simple question. So, as far as I understood, these phlebotomus can spread the leishmaniasis, right? So, so what about the prevalence? I mean, you show the 3 different subtypes of phlebotomus, right? So, which one is the stronger? I mean, the dangerous one.
Can you repeat the question?
Okay, then you identified the 3 different type of phlebotomus, right? So which one is the most significant one? I mean,
yes, Phlebotomus perniciosus is the prevalent, but
okay, then.
Dr. Cavallo?
I don't know.
Can you hear us?
I don't know.
Looks like we have a problem of a connection. So, yeah, sorry, Professor Kim. We are in short of time, so we have to go further. I hope you can understand. So now it's time for Dr. Fadel Charbel. He's going to present research titled Variation of the Pharmacokinetic of Meloxicam between lactating and non-lactating goats. So please, Dr. Fabel, activate your camera.
Mario, just a short message for you.
Yep.
Salwa is per room, is 35, therefore she can speak after this presentation.
Okay, okay.
There was a mistake in the list.
Yeah.
But she is present.
Yeah, okay, thank you. So, Dr. Fadel, the floor is
Good morning. Okay, so as we all know, veterinary medicine is also a form of preventive medicine for humans as defined by public health. Besides controlling zoonotic diseases, are heavily involved in the quality control of animal products and drug residues. Antimicrobials, anti-inflammatories, parasiticides, and insecticides and growth promoters are included. While remains still now the main concern for public health, the emergence of drug-resistant bacteria. Thus, controlling pharmacokinetics and assessing pharmacokinetics is critical not only for clinical application, but also for setting a maximum residual limit MRL and a withdrawal period time for meat and milk. Pharmacokinetics, or PK, is a study of the drug's absorption, metabolism, excretion, and distribution. The problem is that even until nowadays, there's still a lot of data missing on the PK of many drugs in animals. And here comes the importance and the role of us researchers, which imply database and guidelines on the drug's usage. The other issue is that the PK cannot be only assessed in healthy animals in a single physiological state, as the PK of the drug varies according to age, breed in diseased animals, and also depending on the physiological state. For example, due to renal damage, we'll have a decreased excretion, and also due to the heat cycle, we may have an altered metabolism and so on. Thus, my colleagues did previously this study on the variation of the pharmacokinetics of meloxicam between lactating and non-lactating goats. First, to check whether a dosage adjustment is needed in lactating goats, and also to check whether the different routes of administration, whether IV, intravenous, or intramuscular IM, can affect the drug concentration in plasma and milk. Unfortunately, due to COVID and situation, I couldn't present some of my work, which is currently in progress now. So, meloxicam is a widely used NSAID and is a COX-2 preferential. It is used off-label, by the way, in goats. As you can see in this table, we can find all the reported pharmacokinetic parameters. And as you can see in the figures above, the drug was still detected until 48 hours IM and 72 hours IV, with no significant difference between lactating and non-lactating goats. As for the milk and the penetration in the milk, it was still detected until 48 hours, and the penetration was limited and unaffected by the route of administration. In general, the drug penetration into milk can be described using this ratio, which is the AUC milk to plasma ratio, and it depends on many factors like the pH of the milk and the physicochemical properties of the drug. The obtained ratios, which are lower than 1, indicate a higher retention of the drug in blood, in plasma, and a limited accumulation of the drug in the milk. So we don't have a risk of accumulation in milk. And finally, thanks to this study, we know after the calculations that the determined milk withdrawal period of meloxicam is 72 hours, and there's no need to adjust the dose for lactating goats since that the lactation did not affect the PK. And thanks to the half-life, it was confirmed again that the previously used regimen, which is 1 time a day, is okay. And thank you.
So thank you very much, Dr. Chabel.
Thank you.
And Dr. Fadel, sorry, is now time for a question. So looks like there is no.
Yeah.
Oh yeah, Dr. Kim, please go on.
Yeah. Okay, thanks for the presentation. I have just simple one question. So, as far as I understood, you find below the 1, I mean the AUC milk ratio. So, is there any speculation for this low, I mean, ratio for this drug?
Well, we think that the pKa of the meloxicam, which is slightly acidic, around 5.5, 5.6, also affects the entrance of the meloxicam into milk from plasma. So it's a more reversible reaction towards the blood.
Okay, thanks.
So thank you very much. And now I have a note for the board member please, now I'm going to ask to Dr. Havko Salva that belongs to the 35th grade. So for your score, please go up to the 35th or second year, as mentioned in your form that has been given to you. So, Dr. Havko Salva, first of all, excuse me for my bad presentation, and because I didn't call you, I really do apologize, and then excuse me also for my bad spelling of your name. But now is your time, finally, and you are presenting us a research titled Emerging Viral Disease in Swine, Pathological Evaluation of Lesions in Organs of Different Growing Phases in Pigs. So, excuse me again.
Okay. Okay. Can you see my screen?
Yes, perfectly.
Okay, thank you. So, ciao a tutti. Today I will be talking to you about emerging viral diseases in swine. As you all know, emerging viral diseases in general are life-threatening for humans as in animals. And for those of you who don't know, imagine with me COVID-19, for example. Emerging viral diseases in pigs have increased significantly in the last 20 to 30 years. Our research was basically based on 29 subjects from a North Sardinian slaughterhouse, divided into 3 groups, 10 finishing, 10 farrowing, and 9 weaning pigs. 8 organs were taken out of each subject, heart, lung, liver, kidney, intestine, and lymphoid organs, and a total of 232 samples were submitted for histological examination. Moving on to our results. Grossly, no significant lesions were found in the tested pigs, but at histopathology, the situation is different. In order for you to better understand the histological lesions caused by these viruses, I would like you to concentrate with me on the picture in the center. You can highly appreciate the white spaces, which presents alveoli. Alveoli are the actual part of the lung that helps the pig to exchange air and blood and to breathe normally. If you move on to the surrounding 4 pictures, you can see that these lungs are affected. In the first picture on the top left, you can see inflammatory infiltrates, and the white spaces are decreasing, which means we have alveoli collapse. And if you move on to the second picture on the right, you can see that bronchioli are changing their morphology. Whereas in the below 2 pictures, no inflammation whatsoever, but we have high red coloration presenting red blood cells, which means we have congestion. So basically, the infection was graded from severe to moderate to mild interstitial pneumonia. All the tested organs were found to be inflamed, but the most affected organ was the lung. Of interest, 86% of the tested pigs were found to be affected with interstitial pneumonia, and the most affected category was weaning pigs with 78%, whereas farrowing and finishing pigs were 30% and 10% respectively. In conclusion, I think that our results would help to better understand the etiological mechanisms of pig emerging viruses, and can also hopefully can help to control and prevent the diseases in intensive swine farms. And last but not least, ongoing investigation are being held at Zoo Profilattico in order to sequence and detect the presence of these viruses inside the tested samples. And very promising results are ahead. They're very good for my research, but of course not good for the swine. Thank you for your attention.
Thank you very much. Any question from the expert panel board member?
Yes, please. Well, I would like to know if the samples were taken in the slaughterhouse. It means that the pigs were in good health, don't you?
Excuse me?
The pigs were slaughtered in the slaughterhouse, yeah?
Yes,
You take the
yes.
samples of the slaughterhouse, so it means that these pigs were healthy, because if not, they can go to the slaughterhouse, yeah? So they don't have any symptom of the pneumonia?
No, okay. The pigs were healthy. No significant lesions were found grossly, but at histopathology, where we were doing the histological examination, we suddenly found that we have lesions, and we decided that this is severe interstitial pneumonia. But grossly, they look very healthy.
Okay, and the vets in the slaughterhouse, they take these the lambs like they were wood. They can't see anything in the morphological side.
No, not at all. We did not find any lesion morphologically.
Okay, thank you.
You're welcome.
Thank you.
So thank you very much for the question and answer. And now it's time to Dr. Ibrahimi Mohammad Reza to present his study titled Optimizing Injection Time of GSP plasmid into Sheep Zygote. Here you go. We can see perfectly your slide. So the floor is yours. If you can share also your face, I mean your video, would be nice. But if not, you can proceed with your presentation. Thank you very much.
It's still silent. It appears silent. No camera, no voice.
No problem. Go ahead only with your voice.
Ibrahimi, we can't hear you.
Hello?
We can't hear you and we can't see you.
Yeah, he said that his camera.
Yes, but it's not working.
Yeah, but we can hear him.
I think, Mario, that we can skip and maybe we can set Ibrahimi at for.
Yeah. Okay.
Ibrahimi, check your video and your audio, because I can see you here, but we can't hear your voice, and of course we can't see also your face. So you will be asked to do your presentation later. Thank you.
Let's proceed further with Dr. Farinelli Elisa. She's going to present a research titled Direct Measurement of Intracranial Pressure in 2 dogs with brain tumor, poor dogs. So, Dr. Farinelli, please turn on your camera, share your screen. I don't. Dr. Farinelli?
Elisa?
Yes. Can you hear me?
Yeah, we can hear you. We can see you, neither your screen.
Switch on your camera, Elisa, please.
Non riesco a farla funzionare, la videocamera.
Okay, go ahead with your presentation, please.
Okay.
Share your slide.
Okay.
Okay, here we go.
Okay. Intracranial pressure is the pressure exerted by fluids and tissues in the space between the skull and intracranial tissue. The main constituent of the intracranial volume are brain tissue, blood, and cerebrospinal fluid, which are in cross balance. The pressure-volume relationship is known as the Monroe-Kelly theory, which states that since the skull is an incompressible structure, the intracranial pressure is determined by changes in the intracranial volume, and that the increase in volume of one of the components must be compensated by a decrease in volume of another. If this does not happen, the result will be an increase in intracranial pressure. Intracranial hypertension is a cause of clinical deterioration in animals with brain disease, leading to deterioration of brain function and or death. Therefore, it is necessary to detect intracranial hypertension, treat it quickly, and monitor its evolution. The aim of this work is to describe the simultaneous monitoring of intracranial hypertension through direct measurement and ultrasound evaluation of the optic nerve sheath diameter in 2 dogs with chronic neurological symptoms. Currently, the gold standard method for assessing intracranial pressure is a direct measurement using an intracranial probe inserted into the brain parenchyma. However, it is an invasive and expensive method that requires general anesthesia, suitable equipment, and qualified personnel. The indirect measurement of intracranial pressure has recently been described through the ultrasound evaluation of the optic nerve sheath diameter. It is a fast, non-invasive method that does not require general anesthesia and can be repeated over time to monitor intracranial pressure. The optic nerve is accessible to ultrasound because in dogs the bone orbit is incomplete and the dorsolateral edge of the orbit is closed by the orbital ligament. The optic nerve sheath is the anatomical extension of the dura mater, and the subarachnoid space surrounding the optic nerve is in continuity with the intracranial subarachnoid space. It is a thin, curvilinear and hypoechoic structure with linear and parallel edge surrounded by fat and a muscular cone. The increase in intracranial pressure pushes the cerebrospinal fluid into the subarachnoid space with expansion of the sheath that surrounds the nerve. During the study, both dogs underwent neurocranial magnetic resonance imaging that indicated picture compatible with a malignant neoplasm, invasive and responsible for an important mass effect. The direct monitoring of intracranial pressure that confirmed the presence of intracranial hypertension was performed in the operating room, while indirect monitoring was performed before skin incision, 5 and 10 minutes after the end of the administration of the hyperosmolar therapy. Changes in direct intracranial pressure values were associated with changes in optic nerve sheath diameter values, assuming a correlation between these 2 methods and the utility of indirect method in veterinary medicine. More case studies are needed to verify the usefulness of the indirect method for identifying and monitoring intracranial hypertension in veterinary medicine. Thank you for your attention.
Thank you very much, Dr. Farinelli. So it's time for question. Is there any? So it looks like there is no any question. I have a silly one. I'm not a clinician. I'm not a vet. So, but I can imagine that you are a clinician. So, yeah. How many dogs with brain cancer have you encountered in your career so far? Approximately.
My career now, I degree last year and my career is very, very little. But this study is a start work for to continue a big work. But dogs with cancer, we have to do an imaging for to see the cancer, and not all dogs maybe do the MRI, not the owners want to do.
It's too expensive. It's too expensive. Yeah. So thank you very much indeed. It's now time to jump to the next communication from Giagnoni Lucia. Dr. Giagnoni, yes, please.
Good morning, everyone.
There you go. And you are going to present us a research titled Studying the Dynamics of Microbial Communities in Milk and Dairy Products. So the floor is yours.
Thank you. Thermoduric bacteria can survive during the pasteurization of raw milk, leading to a decrease, but not a complete elimination of microbial load. Also, during the successive refrigeration, microorganisms, especially psychrotrophs, can grow, affecting cheese yield and sensory properties. The aim of this study is to evaluate microbial communities in raw milk and during the shelf life of fresh cheeses using DNA metabarcoding approaches. With this aim, the DNA has been extracted and purified by Biospin 96 platform according to the manufacture protocols. Then the quantification of the DNA extracted has been analyzed with qubit fluorometer. The real bacteria content has been assisted by real-time PCR using universal primers. For sequencing, the library preparation will be performed on the region V2, 48, V3, 6, 7, 9 of 16S RNA gene using universal primers. The combination of two primer tools allows for a more accurate identification DNA sequencing based. The sequencing will be performed on iOS5 platform, and bioinformatics analysis will be performed on IO-Report2 in GreenGenes database. The expected results will allow to acquire a more accurate characterization of microbial communities of raw milk and during shelf life of fresh cheeses, to define their evolution over time, to define shelf life determination, to improve organoleptic properties, and to ensure hygienic quality and compliance to food safety criteria. Thank you.
So thank you very much, Dr. Giagnoni. Now the questions are on. So it looks like no question. Well, I have a question. I see from your slide that you have been to Padua to have this research.
Yes, this research is a collaboration with the University of Padua. It's a really recent collaboration, just started this year, and is in collaboration with the industry Nononanni to study the microflora, the microcommunities of raw milk during the shelf life of fresh cheeses, to improve the sensory properties and to improve also the shelf life of their product.
Okay, but are you dealing with milk coming from Sardinia or milk coming from the other region of Italy?
No, it's milk from that the industry Nonodani used, so it's from near to Treviso.
Okay, thank you very much indeed.
Thank you.
So it's time to go ahead with Dr. Maltana Alessio. So please turn on your camera. And Dr.
Can you see me?
Yes, now perfectly. Share your screen, please. Dr. Maltana is going to introduce us a research titled Productive Effect of resveratrol against cadmium-induced toxicity on oocyte maturation and fertilization. So, Dr. Maltana, the floor is yours.
Thank you. 1 moment, please. Okay. My project concerns the effect of environmental pollutants which affect female reproduction. In particular, we have focused on cadmium, which is a highly toxic heavy metal, ubiquitously present in the environment due to the anthropogenic activities. It is worrying for animal health because it has a long half-life and a low expression rate, which leads to a bioaccumulation in reproductive systems. So the aim of this study was to investigate the effect of resveratrol, a polyphenol with antioxidant properties, on in vitro maturation and fertilization of ovinocyte matured under cadmium exposure. So sites were divided in 3 different experimental groups. A control group where oocyte mature under standard condition with hormone and nutrients. A second group where oocyte were mature with the supplementation of cadmium at 2 micromolar concentration, and a third group where oocyte were matured in presence of cadmium and also were treated with the resveratrol at 1 micromolar concentration. After that, matured oocytes were stained and analyzed under confocal microscope to assess oocyte quality markers. In a second experiment, oocytes were also fertilized by spermatozoa and was assessed the fertilization rate. As a result, how can you see here in the representative to control and cadmium resveratrol groups, while below you can see abnormal condition of the quality of the oocyte representative to cadmium group. So cadmium causes oxidative stress, abnormal distribution of mitochondria which are polarized and clusterized within the cytoplasm, also cause abnormal diffusion of cortical granules, and also abnormal distribution of actin filaments, and also you can see here some clusters, abnormal cluster of actin. So resveratrol treatment restores this abnormal condition. As you can see, ROS levels are lower in control and cadmium resveratrol groups. The distribution of mitochondria is homogeneously distributed, and so we have a normal distribution of subcortical cortical granules, which are in the subcortical area, ready to be released after the fertilization of a single sperm cell, and also a normal condition of the actin structure. In the second experiment, in the fertilization, we have seen that resveratrol treatment ameliorates the normal ratio of fertilization by preventing polyspermy. And in conclusion, we can see that this work demonstrates that resveratrol could be taken in consideration for the use on in vitro maturation media. Thank you for your attention.
Thank you very much, Dr. Maltana. It's time for question.
Yes, please. Well, this is beautiful. Just one thing, I don't understand why you call more than 2 ppb percentage. This means the polyspermic
Polar body.
Polar body. Why 2 more polar body instead of polyspermic in the conventional one?
Yeah, pronucleus is an error. Pronucleus, sorry.
It's pronucleus, not PB.
Non polar body.
Okay, because I said the first time that I see too many polar bodies in the polyspermic. Yeah. The other thing, just the control and the control group is exactly the same in the criteria that you have studied, that the group of control plus resveratrol. Yeah, the first this fig. The pictures. The picture is control.
The pictures.
It's exactly the same.
Yeah, it's exactly the same because we have done they are representative to control and carnivore rest group, because in this slide we have not so much space, so they are quite similar.
Okay,
The images are quite similar. Level of loss were very similar, okay, and even distribution of mitochondria and subcortical granules, etc. Okay.
Okay, this is that I didn't understand. Thank you, Alexia.
Thank you so much.
So thank you very much, but I have a very small question from my side as a toxicologist. You found in your conclusion that resveratrol 1 micromolar can give this, sort of safe effect. Do you guess that this concentration is easy to be achieved by consuming cherry, blueberry, or grape, or this natural fruit that contains resveratrol, or we should eat too much amount in order to reach this, and we need a sort of supplementation from different sources?
I don't know, but there will be some further studies to better understand this capacity of resveratrol to get improvement. I don't know, I'm not a toxician, but I think there will be further study to understand it. And now with in vitro studies, we can see these results, but I don't know if in future someone want to take experiment on animals in vivo to better understand the metabolism of resveratrol.
Thank you very much indeed.
Thank
you very much indeed, Dr. Maltana. Let's proceed ahead with Dr. Morrone Sara. So please turn on your camera, Dr. Morrone. Yeah, we can see you. Share your screen while I'm going to introduce your research titled Evaluation of Seasonal Variation of Morphological Parameters of Apis mellifera through the use of 3D models. So the floor is yours.
Good morning. It is clearly demonstrated that seasonal differences occur in metabolic activity of Apis mellifera. In fact, while winter bees can live for over 250 days, summer bees could only survive for 25 to 40 days. Despite this evidence, there's still a lack of understanding about the parameters that determine this distinction. Can they be distinguished according to their morphological or nutritional condition? Well, the goal of this study is to use a real-time, non-invasive technique to examine the morphological variation of Apis mellifera in different seasons, thanks to the use of a 3D model. And to achieve this goal, 12 bees have been harvested from 2 different apiaries on the northwest of Sardinia, in particular from Olmedo and Barrett's apiary, from November 2019 to May 2020. First of all, bees have been subjected to linear measurement. They have been individually weighted, and the length as well as the width have been measured. After that, following that, every bee has been photographed thanks to the creation of a dedicated photo set for a high resolution of small object, and using a software called Agisoft Metashape Pro, 3D models were created. Although the research is still in its early stages, the preliminary findings are promising. In fact, the software could report like an average of 19,825 points for each 3D model. The software could calculate, evaluate in a detailed way the volume of the animals, not only the planar measurement. Furthermore, this model allow us to separate and isolate every single segment, so we can we can measure in a very detailed way the head, the chest, and the abdomen of all the animals. As you can see at the bottom right picture, the model consists in a union of a huge number of points, and they can be evaluated alone, individually, or compared to the others. The software function, it's called wireframe, And this function allows us to the evaluation of every single details of the animal. Finally, the software that we use could create a 3D model, as you can see on the right picture of the bee, that unfortunately I cannot show the animation because the bee can be seen in 360 degrees. And we can create with this software not only from photos of the bees, but also from videos. And that will allow us to a completely non-invasive study and a completely non-invasive comprehension of the difference in morphological condition of the bees in the different seasons. In conclusion, the 3D models could be employed as a real-time monitoring tool to determine the shift in metabolic activity of the entire bee colony based on foraging bee activity. Thank you.
So thank you very much. And Dr. Morone, any question from the audience?
Yes, thank you, Dr. Sara. Just a question about breed. How can you be sure that you are working with a pure breed?
I'm sorry, I cannot hear you.
Yes, my question is about bees breed. How can you be sure about the breed of Apis mellifera that you are working on it? We know very well about the insemination in bees and to have pure breed. And as there are big differences between breed and another, how you can sure about the breed that you are working in for the dimension that it is pure?
So we went to these two apiaries and we take these bees. So we studied a single bee and we photographed the single bee. And after the software allow us to recognize the different type of bee. So we can understand and we can evaluate every single bee from all the details. As I told before, the software can detect many points, and the points allows us to understand the single bee, not only the animal, like the bee. And another interesting tool about the software is that if we can do a video, we can understand every day the difference of every single bee. So we can understand in relationship to the temperature and to the humidity, what are the different morphological conditions of every single bee.
Thank you.
You're welcome.
Thank you very much. I have a small question, Dr. Morone, for you, because your research is titled Evaluation of Seasonal Variation, blah, blah, blah. But according to your presentation, I only saw how this software works. And I didn't hear from you any kind of results that you have found. So
yes, we found some results in planar measurement, and the study is not finished yet. We started this study, and now we have seen that we can do that. We started with the results, but we cannot say a real result because the study is not finished, and we would like to we wanted to understand before how to do a good 3D model, and then every single part have been started to measure, and after we will continue, we will finish the measurement, and we could finish the study and tell everyone if there are differences from planar to 3D measurement, and what are these measurements that now we have just for planar measurement.
Clear. Thank you very much indeed.
You're welcome.
So now it's time for Dr. Cini Maria. So please turn on your camera and
hello to everyone.
Good morning. Share your screen, please, while I'm going to introduce your research. Dr. Cini is going to introduce us her research titled Prevention and Control of Laboratory-Acquired Parasitic Zoonosis. So,
Okay.
Dr. Cini, the floor is...
Can you see my screen?
Perfectly, perfectly.
Okay. In the research field of parasitology, it's very common to daily work with species that could be infecting for humans. So I started looking into available literature, searching for any studies.
However, there are no comprehensive epidemiological data regarding laboratory-associated infection for parasitic disease. The last study dates back to 1976. For this reason, my research work aimed to investigate then the main characteristic of principal zoonotic parasite in order to assess the risk of developing infection at workplace together with the indication of preventive measure. The WHO classified biological agents into 4 groups based on pathogenicity, mode of transmission, and the availability of effective treatments and prophylactic measures. The most of zoonotic parasites considered in these studies, as you can see in this slide, belong to group 2 with a moderate individual risk, while Echinococcus granulosus and Taenia solium are included in group 3 for laboratory workers because they present an high individual risk. In fact, the eggs of Dicertoda can survive for at least 1 year, but are sensitive of chlorine disinfectant 0.5%. Instead, protozoan parasites such as Giardia, Cryptosporidium, and Toxoplasma gondii have an high environmental resistance, but also disinfectant resistant. The structure of the cyst and oocyst reduce the efficacy of solvents and acid. Fortunately, we can prevent this parasitosis through the use of personal protective equipment like gloves and lab coat, and by following good laboratory practice and protocol, one above all, and washing before and after manipulation. This simple measure can avoid, for example, the percutaneous infection by Ancylostoma and Strongyloides stercoraris, but also protozoan parasite infection with an high disinfectant resistance, as I told previously. In conclusion, further studies are required to better understand the environmental resistance of cyst, oocyst, and egg in order to employ best disinfection methods. Last but not least, it is important to close epidemiological data gap regarding laboratory-acquired parasitic zoonosis to better understand the potential hazards. Thank you.
Thank you very much. Dr. Cini, any question on this topic?
If I may.
Yeah, of course, go ahead.
Thanks for the interesting presentation. I want to just ask a simple question. So, what is the first priority to secure laboratory-associated infection prevention in our daily life or, research life?
Okay, as I told previously, there are many preventive measures, but the important are disinfection method like chlorine disinfectant 0.5% and washing before and after, because protozoan parasite like Giardia and Cryptosporidium can survive with a disinfectant. So if we wash the hands, we prevent the disinfection.
Okay, then thanks a lot.
Thank you.
So thank you very much, Dr. Cini. And now the last speaker, we are going to call back Ebrahimi Mohammad Reza. Okay, now we can see you perfectly. Good morning.
Good morning, everyone.
Good morning. Please share your screen and present us your research titled Optimizing Injection Time of GFP plasmid into sheep.
Yes.
Thank you very much.
You're welcome. Hello again, and sorry. Okay. Sorry. Hello again, and please apologize. Please accept my apologize. So, sorry again. I have to stop. Okay, I will go ahead with this. Okay. My project was about optimizing injection time of GFP plasmid as a reporter gene into sheep zygote. As you may know, there are not enough data about the microinjection procedure for introducing the exogenous DNA to the sheep zygote. So we decided to do this experiment to improve our knowledge about the microinjection procedure and to find the best injection time of exogenous DNA to produce transgenic sheep. To do this, as you can see here on the left and the side of the graphical abstract, we collected the COC from the we collected the ovary from the slaughter house and then transferred them to the laboratory. In the laboratory, the COC isolated and subjected to the maturation and in vitro fertilization media. After that, the GFP plasmid as an exogenous DNA was injected to the cytoplasm of the zygote at different time interval, as you can see here, from 6 to 8 hours post-fertilization to 12 to 14 hours post-fertilization. Then the injected zygote transferred to the in vitro culture media for 7 days and then evaluated for GFP expression. However, the result, was not very good in the blastocyst rate because of the embryo manipulation. But the main result indicated that the injection of the GFP plasmid at 8 to 10 hours post-fertilization is the best time for expression of the GFP in the sheep zygote. This means that at this stage, at this time, the oocyte components are ready to accept the exogenous DNA and probably for integration to the host genome. This result can be applied for next experiment to produce transgenic sheep. Thank you for your attention and sorry for problem. I am at your service.
Okay, thank you.
Thank you very much. Any question?
Yes, please. You know if the green protein was in the pronucleus?
Sorry, would you please repeat again?
Yes, the injection. The aim of the injection was the cytoplasm around or in the pronucleus?
Well, no, it was a cytoplasmic injection, but we tried to go ahead near to the pronucleus. We didn't disturb the pronucleus. Just we tried to go ahead as much as possible near the pronucleus. But generally, it was the cytoplasmic injection.
Yeah, because your idea is that close to the pronucleus, the protein can get into the nucleus.
Exactly.
Wow.
You're right.
Okay, because your protein is covered with something special.
You're right. You're right. But, according to our experience, if we, inject to the pronucleus, the blastocyst rate will be very low, and we could disturb and we could kill the embryo. So we decided to inject in the cytoplasm, but near the pronucleus instead of going ahead to the injecting to the pronucleus.
Okay, that's it. Thank you.
You're welcome.
So thank you very much indeed, Dr. Ebrahimi.
Yes.
And now the presentations are over. Thank you very much for all the presenters, the attendants, and the commission. And I would like to give the words to, I can see even Antonio. Antonio, you want to say something?
Yes, I would like to say that there is a little bit change in the program, and we will postpone the hours. And before we give words to Professor Fiammetta Belliware for explaining the available position for this year. Okay, so please everybody stay online, and after Fiammetta there will be the awards ceremony. Thank you.
Thanks, Antonio, and congratulations to all of our students. They were really very interesting presentation, very well done. And so we'll be waiting for the results of the evaluation by the jury soon. In the meantime, I wanted to just introduce you with the next call for the admission to the PhD course in veterinary medicine, which is now open. As you saw, the PhD course in veterinary science of our university covers all the aspects of veterinary science, from physiology to surgery, pharmacology, but also internal medicine, pathology. And so we really cover all the aspects of the veterinary research areas. The course will then last 3 years after admission, and students during these 3 years courses will have to follow also some teaching activities in lectures or seminars, besides carrying on their research program under the close supervision of their tutor. During this period, motility is strongly encouraged because we really think that internationalization, and especially in research, appear abroad open your minds and gives you more expertise and ideas, and also project you toward an international network for your postdoctoral, if you want to continue in the research field after your doctoral degree. And so mobility is also funded by the University of Sassari under the program Erasmus and Ulisse, and you will be given a 50% increase of your salary during the period of mobility. Also remember, please, the opportunity given by the collaboration we have with the new University of South Korea, and here we have Professor Kim, and of the University of Barcelona, with which we have a double degree program. So as I just told you, the call for the admission at the 37th cycle is now open and will close the 1st of July. So if you are interested, please hurry up and give a look at the link and the site where the call is posted. It is just one call because the PhD program is coordinated by the School of PhDs of the University of Sassari. So there is one call for all the PhD courses of the university. And the selection will be completed by August, and after that enrollment will be completed by October. And so the course will start the 1st of November with the beginning of the academic year. The selection procedure, you will see if you want to have a look at the call, is really tailored to recruit just young postgraduate students, and no specific experience in the research field is required, because we just want young researchers which are strongly motivated and which have a strong attitude toward the research and study. And this is why the call is not restricted to any specific degree. So to enter the PhD program in veterinary science, you don't have to be graduated in veterinary medicine. You just have to be graduated in any discipline. Of course, scientific ones are usually preferred, but there is no restriction for the application at the regards of a specific degree. After that, the selection procedure foresee 30 scores for your curriculum, and then 70 score for the interview. You don't need to submit any research project to apply for the course in veterinary science, but just your CV. And we will evaluate your CV, especially for the final grade, for example, so very general terms, and also for the public number of publication, maximum 5 publication will be considered, and our courses of qualification. And we will give a 4 points score more if you have a B2 level in English. But you can also apply even though you are not graduated, but if you plan to graduate by October, because you can apply the sub condizione. So you will declare that you will graduate by October, and you can also apply even though you're not graduated at the moment. So this is the general graduation of the titles of the CV. After that, there is the interview. During the interview, which will be scheduled by which will take place orientatively late July. Jury will evaluate the knowledge of the English language, for example, because part of the interview can be taken in English language. Your motivation, so your attitude and your motivation toward the research, which is really a key point in a PhD research program. And also specific knowledge and skill in one of the topics of the call, which are listed in the Annex 1 of the call, right at the end of the file. You will find the veterinary science, all the information on our course in veterinary science, with the aim and of the course, all the area of interest, the position open for this year. The course is divided into curricula, one in food production, quality and safety, and the other one in reproduction, animal pathology, breeding and welfare. So we have 2 positions in the first one and 4 positions in the second one curriculum. You will find all the detail in the Annex 1. And also we have also prepared short videos with all the tutors, so you can also kind of know them, and they will be posted in Facebook in the next day. So you can also have a look at the position and the research program that will be carried on after the selection. And please be aware that the additional position can be also added, because we apply to other research program, international research program, and so other position can also came out by the end of the summer. And but you have to apply now, because we can recruit only the students selected by this call. So during the interview, we will also ask you to briefly present a research project on one of the topics that are listed in this annex, and cover from the food production area to the histopathology, internal medicine, and also parasitology. And the commission will particularly evaluate the knowledge and the skill in the communication and the attitude toward the research. So please apply. There is really no specific title required, just a strong motivation and attitude toward the research and an interest in the field that are open this year. I think that for the call, if you have any question, you can always contact me. There is my email address. You can easily find in the internet, and Antonio will also post it in Facebook in the next day, so that you can easily have access. You can write me anytime for any further information. So we're waiting for you. Please apply. And I'm done, Antonio.
Thank you, Fiammetta. So we are waiting the jury. I think that we have almost decided everything. Mario, are you online?
Yeah, I'm right. I just have to ask you just 1.5, at most 2 minutes, if you can.
Okay, I think that
I will get back to you with the declaration directly from the jury.
Okay, do you need to speak with Fiammetta too? I will continue with some minutes with the students here.
Yes, please, thank you.
Okay, Fiammetta, I will take the floor. So thank you. Just to break the time, in the meantime that we get the judge of our jury, just few tips for our students. I have seen your presentation, and by the communication point of view, I think that almost of you score very good. Most important, some of you try to don't read during presentation. This is very appreciated from people. Some of you also try to smile at the beginning, at the end of your presentation. This is also a good tip. And some make some small errors that could be modified during time, making some exercise. The first thing is try to connect from a good place and have a good connection, because your face is more important sometimes than your contents. And even when you have very few minutes like here for making an elevator speech. Therefore, this is very important, and for this reason I asked you to switch out your video, and it is important that the commission see you. And then I think that's all, and we will of course repeat this contest also for the next year, and maybe we can try to shorten the timing, because as I told you to some colleagues of my age, the life is fast in new media age. I don't know if there were anybody here that would like to say something. No. And also take the position of Mario for telling you that all posters have been evaluated by your colleagues, and we have an awards for Anna Jagu, that scored as the most voted poster in our poster session. There is only an awards, no prize, and it was also pity because I have seen Anna Jagu performing last year in a contest presentation, like an elevator pitch, and probably you should have a lot of chance to win this year, and so waiting you for next year for presenting your work. Anna, are you here? I can see you.
Yes, I'm here.
Why do
I
you
had
why,
a
why,
very busy time.
why don't you present your work?
I was busy. That's the answer. So next time will be better.
Okay, perfect. And
I'm ready if you wish.
Okay, yes, I have ended my arguments and also present the poster session, and the floor is yours, Mario.
Thank you very much. So it's time of announcement now. I would like to ask Professor Chadi Hoshri to announce the winner for the first year, or well, mention the 36th year. So Professor Hoshri.
Yes, thank you. After evaluation, I would like to congratulate Dr. Alessio Maltana. Dr. Alessio Maltana, the first year is the winner.
So thank you very much, and congratulations to Dr. Maltana Alessio, that presented the research titled Protective Effect of Resveratrol Against Cadmium-Induced Toxicity on Oocyte Maturation and Fertilization. Thank you very much indeed. So now it's time to announce the second year from Professor Maria Teresa Paramio. Please go ahead.
Okay, I would like to congratulate to Frau Adel for that she is the winner of this second year. Congratulations.
Thank you. Thank you.
Thank you very much to Professor Paramio and to Dr. Frau that presented her nice research titled Can Antimullerian Hormone Predict Reproductive Life in Dairy Heifers? So, and now it's time for Professor Kim from Tuna National University to announce the winner for the last year, so the 34th grade.
Okay, then, yeah, the winner of 30 years is Dr. Irene Sartini. Congratulations on your success, and congratulations to you all. Thank you.
So thank you very much, Professor Kim, and congratulations to Dr. Sartini that presented her work titled Pharmacokinetics of doxycycline in the goose.
Thank you.
let me spend a few words, if I can, as the chairman of this section. I'm very glad to have chaired this section. I want to remind you that in this PhD contest, the student had a double role. The first one was the presenter, so the main one, but the second one, that is not negligible, was also to reviewer, because they review a poster of their, how can I say, PhD student. So I want to thank all the committee member board, the team that, the PhD team that worked hardly for this contest, but as well as all the PhD member staff of University of Sassari that agree and supported this concept of new PhD evaluation. So maybe Fiammetta, you want to add something to close this contest?
Thank you, Mario. I want to congratulate with all the students, and particularly with the winners, of course, of this contest. We will keep on organizing this kind of event, because we really believe that it is really important for you to get opportunity for public speaking, and you really did a great job. Also to illustrate all the variety of area in which research is carried on under the PhD program. And then I really want to thank you, our super jury, the international jury, Professor Paramio, Professor Kim, and Professor Osri, for their work and commitment also, because it was also in the last week hard work to organize everything. So thank you very much. I truly believe that our collaboration under the PhD program for us is really important, and this is also a way to foster it, and we are available, and please keep in touch and keep on collaborating with us. So really, really thank you for your effort to everybody, and also to Mario for to be a great chairman, and to Antonio Varcasia for organizing all the events, also the live Facebook, and to the PhD board, in particular Gisela Bogliolo, Maria Grazia Cappai, and Raffaella Cocco, which helped us in organizing this event. So and also for the next, which I hope they are following us, the next student of the PhD program, please apply and get involved with this program. It will be really a good opportunity for you. This was like a way also to get to know each other. Okay. So I am done.
Very nice experience. Thank you, Fiammetta. Very interesting. Very interesting. I think I'm going to copy this experience for my university too.
We can hope that the next year we can do sharing together the same contest. You know, we are sister universities, so why not? We can gather our PhD and do a bigger contest. Maybe same structure, but with the highest number of students. Why not?
Yeah, this would be a very good idea, Mario. With all the students from the 4 university, would be great.
Okay, thank you very much indeed.
Thank you. Thank you.
Bye-bye.
Thank you. Bye, everybody.
Thank you very much.
Bye, everybody. Thank you.
Bye.
Thank you.
Bye. Thank you.
Thank you.
Bye.
Bye. Thank you.
Bye.